Hi, we are going to present to you the practice 5 of diffusity of a mosquito repellent. The objective of this practice was to develop a protocol or a methodology to determine the effect of the temperature on the diffusity of a mosquito repellent. What are mosquito repellents? Mosquito repellents are volatile chemicals that, when are applied to the human skin, repel mosquitoes in the opposite direction from their source, discouraging contact and biting. What determines the efficiency of a mosquito repellent? The effectiveness of mosquito repellents is determined by their diffusion coefficient. This parameter can be considered one of the crucial physical properties of repellents that can affect their effectiveness against mosquito bites and can be an indicator of the release rate of the repellents. This depends on the vapor pressure at the room temperature and is associated with the boiling points of the repellents. To evaluate this diffusion coefficient of the repellents, we are going to use spectrometry analysis with UVVs. What is the diffusity? The diffusity is the movement of molecules of a substance from one area of higher concentration to another of lower concentration. What is the importance of evaluating the effect of temperature on the diffusity of a repellent? As I mentioned before, we are going to use a spectrometry analysis to evaluate this effect. It is important to carry out this study since personal protection against mosquitoes through the application of repellents has become useful practice that can reduce or prevent the transmission of insect-borne disease by analyzing the efficiency. In this way, compounds that have lower boiling points may allow better vapor repellency but may dissipate more quickly. Compounds with higher boiling points have a low pressure and will be ineffective at repelling over a predefined distance. This can allow mosquitoes to land but not to bite.
Now we'll talk about the methodology. First, we'll talk about the methodology used in the lab. So first, we add the food colorant to the mosquito repellent. Then we take 4 jars and label them with the temperatures 10, 30, 60, and 90 degrees Celsius. Third, we mark a height H in each jar, and it must be the same. Fourth, we add 30 milliliters of water at the temperature that corresponds to each jar. Fifth, we add 10 milliliters of the repellent to one jar and start to measure time. Sixth, every minute we take a sample at the height H and store it. This for 4 minutes. And last, we repeat steps 5 and 6 for each jar. Finally, we analyze the concentration in the UVVs. The methodology that we proposed was to first take a tube that has a 2 millimeter diameter. Then we covered one end of the tube. We marked the height. Fourth, we add the repellent up to the height that we marked. Fifth, we take data of the new height as it evaporates every few minutes. Sixth, we graph h versus t squared. Seventh, the slope is the approximation of the diffusivity. Eighth, we repeat the experiment with different temperatures.
Results. First, we graph the results of absorbance versus wavelength, and even though the highest absorbance varied between temperatures, they all peaked at around 490 nanometers, which means they all have the same molar extinction coefficient with a value of 0.04 liters per mole per centimeter. The behavior of the diffusion coefficient against the temperature shows an increase in the coefficient with a higher temperature. The graph of diffusion coefficient versus concentration shows no apparent behavior. This is likely due to the errors throughout the experiment and/or insufficient data points to reach a conclusion. Now for the analysis of results. We're going to start by talking about the limitations of the simplifications made for the Serret equation. These simplifications were stationary state, This restricts us from using Sauret's equation for systems in motion. There is no temperature change inside the control volume, and thermodynamic coefficient gamma is neglected, and there is no thermal exchange between the species. These two simplifications neglect the effects of temperature and heat exchange, making the results inaccurate. In spite of these limitations, the simplifications we used allowed us to use Sauret's equation to calculate the diffusion coefficient. Fick's law of diffusion is the most common equation used to find the diffusion coefficient, as it describes the movement of molecules from a higher to a lower concentration region on a larger scale. This law can't be used on a molecular scale. This is because it describes linear and constant movement of molecules. On a molecular scale, molecules do not behave in such a way. They move randomly and in all directions, called the Brownian movement. In order to be able to use said equations, we must be able to determine the molar flux, which can be particularly difficult with the equipment at our disposal. You might be wondering, how does the temperature affect the diffusion coefficient? A higher temperature usually leads to a higher diffusion coefficient because of the increase in the molecule's kinetic energy and therefore its movement. This can be seen in our graph from 30 degrees Celsius to 90 degrees Celsius, where the diffusion coefficient increases with the increase of temperature. The significance of the diffusion coefficient and what does it actually mean? The diffusion coefficient is the amount of particular substance that diffuses across a unit of area over time, under the influence of a concentration gradient, from a higher to a lower concentration. A larger coefficient translates into a wider area reached by the substance every second, or a faster diffusion of said substance in a controlled volume. The effect of the composition and presence of various chemical compounds on diffusion. According to an experimental study, diffusion slows down when the volume fraction increases. In this way, the diffusion time also depends largely on the molecular composition. This is because the average distance between the macromolecules becomes shorter, effectively rendering the system denser and making it harder for the molecules to diffuse.
Now for the conclusions. First, the objective was completed since the diffusivity was measured successfully with the protocol that we use in the lab. Second, the diffusivity first decreased as temperature rises but then increased due to molecular interactions. This was similar to the example that we saw in class with methanol in water. Third, there was also seen a correlation between the concentration and diffusivity. And last, we can only see the magnitude of the diffusivity since the calculations were made with the simplifications in the formula. Here we have the references used. Thank you.